So this is 3.22 food processing and spoilage. So the first question kind of asks you to the stages of the reduction of yogurt. Okay, so you want to heat the milk and add a bacterial culture. Lactose will turn that into lactic acid. The acid curdles the milks and lowers the pH scale. See if the sample's ready, and then you kind of like add the flavour in, add the colour in, stuff like that. Next question then asks you to plot the data on a grid. Okay, the only other thing it says is to join the points, which is really rare in science. Okay, to ask you to join points and not do like a straight line or something. Okay, do the results suggest the claim that bacteria will not survive in the ants in the stomach? The results are up and down, hard to say if they would survive or not. Do the results agree with the manufacturer's claim that the number of bacteria in the stomach will increase? It increased on day 2 and 4 after drop-in, so again, a little bit hard to make definitive conclusions. State 2 methods of making sure this would not happen in harmful bacteria. We also pasteurize and we add that bacterial culture to lower the pH. One symptom of food poisoning caused by harmful bacteria is sickness. You could have had diarrhea, temperature, stuff like that. Another species that can cause food poisoning is an E. coli, so I put salmonella. Okay, again, same question there about making yogurt. So heat milk and add bacterial culture. Lactose will then be added to the lactic acid. The acid curdles the milk, lowers the pH, the sample is ready. Again, add the flavourings, kind of food colouring if you need to. Okay, nice 6 marker here describing pasteurization and homogenization. Pasteurization kills the bacteria by heating it to high temperatures. The milk is heated to 72 for 15 seconds before cooling to 3. Okay, so that will kill any bacteria. Okay, homogenization takes place by pumping milk at high pressures through narrow tubes, breaks the fat into smaller droplets. Fat floats to the top to be removed. It is used to reduce fat content, making skim milk and semi-skim milk. So depending on how much fat is removed, depends on what type of milk you want. Describe the optimum conditions required for food spoilage. It needs to be warm, moist, and they need a food source. Explain one way in which food spoilage can now be slowed down. Put it in the fridge to cool to cold for so the bacteria could not. Put in a fridge. For bacteria to stop growing. Maybe I was trying to write cool down like that. Okay. 3 hygiene rules that you could follow to make sure that you are keeping clean. Protective clothing, sterilisation using... I don't know what's happened here, so I'll just read them off from the side. So, protective clothing, sterilisation using high temp, good personal hygiene. Name 2 other microorganisms that cause food spoiling, E. coli and salmonella. Okay, state 3 conditions needed for microbes to grow, moist, warm, food source. You can see the questions are very similar throughout the bank of questions that we have to give. Why is equipment sterilised? To ensure that no other bacteria is growing. Okay, this bit at the top was just the method for making aseptic technique or carrying out aseptic technique. Why is the lid on the Petri dish only lifted a little so no airborne bacteria enters? Why is the Petri dish stored upside down? Stop condensation, because they do respire, so that the air will rise, and if you store them upside down, then it won't fall on the actual agar plate. Why is the Petri dish incubated at 25? Any higher and it will be temperature and the harmful bacteria will grow. Okay. How can you improve the method and why? I would put slightly open the lid of the Petri dish, remove the lid of the bottle containing the bacteria, slightly open. This needs to be put in the Bunsen burner first. Okay, put in the Bunsen burner first. Use some of the inoculating loop to remove some of the bacteria from the bottle. Zigzag to spread the bacteria over the agar plate. Put the lid back on the Petri dish. Put the Petri dish into an incubator at 25 for 24 hours. You could have put to 48 hours there. Okay, it's all about ensuring that it's really nice and clean and nothing can affect the growing bacteria. Final question then is looking at bacterial growth. Okay, the first thing I asked you for 4 marks was to plot and do a line of best fit. For me, when I was doing it, that point didn't quite fit in my line of best fit. Okay, depends how you did it. Describe how the number of bacterial colonies changed over time and how this affects the storage numbers. Increases over time, higher temperature means more bacterial colonies will grow. Okay, 10 to 25 then. So you're looking at 10, which would have given you 4 bacterial colonies, 25 would have given you 6, so I took them away from each other, gave me 2 colonies. 50 colonies to start, there's going to be 50 colonies to end. That's a really nice 1 mark question there.